Well, thank you very much for inviting me, and good afternoon, everyone. I'm going to be talking about the quality assurance of vaccines, and I'm from the National Institute of Biological Standards and Control, or NIBSC, or as we call it, NIBS. We miss off the C at the end. It does rile me a bit when people try and fit all of the letters in, say NIBS, but never mind. What are the principles of the quality control of vaccines, and why do we do it? The principle of batch release process is what we call it, is that the clinical trial batches which have got proven safety and efficacy, and everything which is produced subsequently to those clinical control batches, the safety and efficacy can be assumed if you can demonstrate that those batches do not differ significantly from the batches which are used in the clinical trials. So this is what everything that we do, this is the principle which underpins it, that the new produced batches of vaccine have to be the same as the ones used in the clinical trials. So how are pharmaceuticals assessed? There's obviously the manufacturer reliability, which is the GMP, or the good manufacturing practice, which ensures that the product is produced consistently. There's the characterization of the final product. There's also the clinical trials, which obviously you probably had a lot of talk about today. And then there's the continuing sort of pharmacosurveillance and inspections of the sites where they're made, and the post-marketing surveillance as well. So all of these things are put in place to ensure that the vaccines are safe and efficacious. I'm going to talk a little bit about licensure, just so you can place where NIBS actually works and how we operate and how we interact with all these agencies. But in effect, if you have a new vaccine which comes to wants to go to market, there's two possible approaches. Now this is a bit of a slimmed down diagram. There's obviously quite a lot of European bureaucracy which goes on here, but the two choices are, at the minute, they can either go through the centralised procedure, which is a European-wide process, which is conducted by the EMA, of the European Medicines Agency, and then from there the marketing authorisation, which is the document which lays down all the specifications about the end product and how it should be produced, and the specifications associated with its consistency. Or alternatively, the company producing it could go to the National Licensing Authority, in which case it's the MHRA, and then after it's been authorised by the MHRA, they can issue a national marketing authorisation, so the vaccine can be sold in the UK in this case. But also this authorisation can be mutually recognised throughout the whole of Europe, so it would be possible to go through a national licensing authority and then have it mutually accepted through the rest of the countries. So likewise, a new vaccine product could go through the French regulator, and then it could end up coming into the UK via that route. But there are some other variations on this, but I'm not going to go into all of those. Once the marketing authorisation has been licensed, or a product's been licensed, then the other side of the coin, in effect, is that it goes to the EDQM, which is the European Directorate of the Quality of Medicines. Now, they're not part of the European Union as such, they're part of the Council of Europe, which are the two different bodies operating in the European Union. The EDQM are responsible for looking after the European Pharmacopoeia, which is a document which lists all of the specifications associated with different vaccines and different products, not just vaccines, all pharmaceuticals. And also they control the OCARBA guidelines, which are the official control and batch release guidelines. Now these are the guidelines which are set out for all of the testing laboratories like NIBSC, for which tests that they have to do to ensure that they are testing the safety and the efficacy of vaccines. And as I said, that this feeds down into the OMCL network, which is the official medicines control laboratory network. And each country in the EU has a network member, although not all of them will actually test vaccines. Only about 16 actually test the vaccines. And in the UK, this responsibility is split between NIBSC, which deals with the biological products, so vaccines in particularly, as well as blood products and other biologicals, and the laboratory for the government chemist, which deals with all the chemical products. it's quite funny really that, well, I'll come on to this in a minute, but the LGC comprises about 30 people, where NIBS is 10 times that, even though the LGC probably handles more products than we do. Just goes to show how much simpler the chemicals are compared with the biologicals. So the differences between the biologicals and the chemicals are quite striking. Biologicals are structurally more complex, as you've probably been hearing today from the vaccines, quite complex mixtures, as opposed to a chemical which can just be synthetically produced. Biologicals tend to be inherently more variable. The processes by which they're made often requires the purification from other growing organisms, as opposed to just, as I said, chemically synthesizing them. The biological effect may be impossible to link to the chemical structure. So the actual vaccine itself, just by actually looking at the structure, you might not be able to get an idea of whether it's actually going to, if the structure affects the function or the toxicity or the safety profile of that. So that's a big difference. So in order to be able to assess a vaccine, we actually have to have a standardised bioassays. The bioassays in themselves are assays which involve enzymatic reactions, involve using cell lines or looking at a biological process to monitor the safety or the efficacy of the vaccines or the products. And these assays are, again, much more complicated to run and a lot more difficult to standardise. And also the biologicals also may have other unknown biological contaminants, such as viruses and endotoxins, as opposed to, again, just to synthesising a chemical. It's a lot more straightforward. So because of the difficulty in production and also monitoring the biologicals, the government in its wisdom decided that they needed to set up a special body to look after biological products, especially as there's been quite a large increase in biological products over the years, and also an increase in how complex the biological products are over the years. So the government in their wisdom decided to set up an independent body, which was NIBSC. So NIBSC started work in the early 1970s as and the Biological Standards Board was set up, and that was at the National Institute for Medical Research, which was in Mill Hill in North London. In 1987, NIBS, as it is now, moved to the very glamorous setting of South Mimms. As you may know, if you're familiar with the name, there is a service station at South Mimms, and contrary to popular belief, we are not actually working in the service station. Although the director has looked at diversifying income streams and possibly offering our staff car park for overflow lorry parking. As a part of the process of reducing the number of quangos, or non-departmental public bodies, NIBSC was moved into the Health Protection Agency in 2009, and as you're probably all aware of, in the end of this month, the HPA is going to change into Health Protection England, and NIBSC, unfortunately, will be saying goodbye to the orange ring of trust and hello to the all-seeing eye of the MHRA. But we believe that the fit with the regulatory part of the MHRA and the work that we do at NIBS is actually quite a good one, and it's also one that we see throughout Europe. The Paul Ehrlich Institute in Germany has both the regulatory and the analytical part under the same building, and hopefully it will lead to some good collaborations between us and some improvements. NIBS at the minute employs about 300 staff, the majority of which are all scientists or have come from scientific backgrounds. So what we do at NIBS, the NIBS remit is really split between control and standards. The control is what I'm going to talk to you about in a minute, which is looking after the control of biological products. But the standard side of us is just as big. We are the world leader in the production of biological standards and reference materials. We produce 90% of the world's international standards. These are standards and reference materials for running bioassays. They are diagnostic reagents. They are standards for all sorts of assays, as well as also assays for analysis of vaccines and other products. All of our standards and control work is underpinned by research and development. We are in quite a nice situation is that we're not manufacturers and we're not universities, so we tend to work with both. So we have a lot of research collaboration with actual manufacturers when they're introducing new products, and also with universities, which we try to tend to a lot, and we've had fruitful collaborations with Andy before. We also offer advice. This is advice to both the manufacturers when they're trying to bring new products to market, or new products into developing new products. We provide advice to the EMA and also the regulators on the control of new products. We provide advice to the WHO on their vaccination programs, and we're involved in both the improvement of public health in both UK and the rest of the world. So these are the sort of products that we test at NIBSC. You can see that they're broadly split between bacterial and viral vaccines. Looking down the list, you're probably all quite familiar with a lot of all of those. Perhaps the big emission that we don't actually do any batch release from NIBSC is the pneumococcal conjugate. So we don't do Prevnar. That's done predominantly in France, although it is split between France and Belgium as well. But everything else we've got there, and you'll see that we do have the new MenB vaccine, which is going to be coming through NIBSC. But as I said at the start, I didn't say that I work in the bacteriology side, and particularly work on the meningococcal and the pneumococcal vaccines. We have a number of viral vaccines as well, and we also work on a lot of the paediatric combinations, so all the DTaP, Hibs, IPVs. We also have products which are not quite at market yet or in early stages of development. So they're malaria, C. diff, group B strep, as well as the new combinations of the paediatric vaccines. So we do get a lot of this before it goes into trials even. Our statutory role at NIBS is as the official medicines control laboratory for the UK, which is designated to us by the MHRA. We have batch release responsibilities where we have to conform to the EU law. We also do quite a lot of work on plasma pools as well, and blood products. We're a member of the OMCL network. We also partake in product monitoring, which is post-marketing monitoring, and we have very close relationships with the MHRA, the EMA and the EDQM. We also do the WHO pre-qualification testing. So this is a very similar process to the testing that we would do for Europe, but it's doing for vaccines which are seen for the rest of the world outside of Europe. So we help there. And the testing under Article 58, which is basically the same. These are products which are seen throughout the rest of the world and not necessarily in Europe. So this is a quick schematic of what we actually do in terms of batch release. It's split into two halves. The top half deals with the receipt of the sample. So this is the actual final product. So this is the same product that goes out to the clinics. We receive that sample. This goes off for some tests which we do, which is predominantly looking at the safety, the identity and the potency. And then we have our results which are fed into our database. We also receive the manufacturer's protocol. The protocol is basically, it's the list of the manufacturers, all the testing that the manufacturer has done and their results and the specifications, and we have to review all of that data just to make sure the manufacturer is doing what it says it should and all their results are passing. And so that's reviewed. All this goes onto our database, and then we release a certificate of release of that batch to the marketing authorization holder, and then they're free to go out and distribute it to the rest of Europe and sell it to whoever wants to buy it. We also pass on this information to the other OMCLs in the network, and it also goes on to a central European database which is accessed by all of the network and other people as well. So we usually on average test about 2,500 batches of vaccine a year. One of the questions I'm hoping that everyone's going to ask at the end is how big a batch is, how long is a piece of string. Batches can vary in size quite from 1,000 up to nearly 100,000. We've been involved in the Manafravac project, which I don't know if anybody's, it's giving Men-A conjugate vaccine to sub-Saharan Africa, and the batches of that have been enormous, and they've vaccinated an incredible number of people in Africa. We have targets for turnaround time, so how long we can do to test these, because the other question is, well, why is NIBS always holding up vaccine release? The manufacturer's got it waiting, it comes to us, and then we sort of sit on it for a long time, and eventually we say, okay, you can go away and use it. We do have timelines which we have to stick to. 60 days is what we're statutory target, although we do have lesser targets for other things such as blood products, 30 days. We were the first OMCL to have a formal quality system, and that sort of underpins all our activities. So the other question we always get asked is, well, why do you repeat the manufacturer's tests? The manufacturers spend an awful long time testing their product to at every point during its manufacturing process. All of these tests are done. Why do you need to repeat them? The main argument is that vaccines are given prophylactically to large numbers of healthy people. Therefore, the assurance of safety is absolutely paramount. We have to make sure that these vaccines are safe. It's in no one's interest to make defective products, but mistakes are made. People who work in very, very large industries are under a lot of pressure, and sometimes people do make mistakes. And we are there to make sure that those mistakes aren't carried through to defective products reaching the market. And also we have different analytical methods which can test for the same thing, and some of those methods are able to identify subtle changes in the product, and this can highlight changes in the product, and we are able to report that back, and we usually have a discussion with the manufacturer at that point, and then agree on if anything has changed or what can be done about it if it has. And also we have the batch release as well. It is also a partnership, really. It's not just about us scrutinising exactly what the manufacturer is doing and checking them all the time. It's a process where we're working together. So we're not only just looking at the results, we're monitoring trends of the products, we are developing new assays, we're developing new standards and reagents for these assays, we're reviewing the specifications for these assays. Usually after a new product is brought out, the specifications can be quite wide, and then during time as we generate more and more information on them, the specifications can be narrowed. And also we're introducing new reference materials and standards. Quite often when a standard or a reference is changed, then we can see a change in the actual results we get, and it's our job to monitor those trends and ensure that when references and standards are changed, that these things are kept. So what tests do we actually do? As I said before, the tests are split up into identity, potency and safety. Identity is pretty straightforward, really. It's just making sure that what's supposed to be in the vial is in the vial. So this is done chemically or immunologically, where we have enzymes, antibodies which will detect what's in there and tell us what's in there. Potency is probably the most difficult for us to do. Potency tests can be related to things like toxin neutralization, where we would look for like tetanus, where we look for the production of a neutralizing toxin and measure that, neutralizing antibodies to the toxin and measure those. There can be physicochemical analysis, where we would actually, where there's been a link to the physical nature of the vaccine to efficacy, we can just measure that. So my example of this would be, say, Men-C, where there's the size and the quantity of the polysaccharide component in the vaccine, which is conjugated to the carrier protein, has quite close links with the efficacy of the vaccine. So as long as we can measure that directly, then we can have a good idea of the efficacy. We sometimes have to do immunogenicity, where it would go into a model of some kind, or we can measure enzymatic activity in some of these products, but not all of them. That tends to be more with the blood products rather than the vaccines. And then finally, safety are the other tests we do. So these can be just direct measurements of contaminants such as endotoxin. There could be measurements of pyrogenicity, toxicity. We also look at the purity of the products to make sure that there isn't anything there which there shouldn't be. There can be tests for the histamine reaction, and also just plain good old-fashioned visual inspection, just looking to see if we can see any problems with it. This doesn't happen very often, although on occasion we do find a few things, see a few things floating around in the vials which perhaps shouldn't be there. So what happens when things go wrong? I'm pleased to say that nothing really has gone wrong for quite a number of years now. In fact, 50 years when there was a few problems with polio. But we have to be very careful because, as you all know from headlines which come out in the tabloid papers, that the public is very suspicious. And if there's, they will pick up on the slightest inkling that there may be something wrong with any of these products. So we have to make sure we operate with a lot of integrity. The number of people who are producing vaccines is also increasing rapidly, and it's also increasing in where they're produced around the world. I think the majority of actual chemical products are actually produced outside of Europe these days, most of them in either India or Bangladesh. There's also a very large number of manufacturers coming from China. And while we're not saying that they're unscrupulous, they certainly haven't got the experience of the big manufacturers from Europe and in the United States. The OMCL network in itself provides a communication route for information on batches which are withdrawn. Now, these are batches which have been withdrawn through testing, so they have never actually got to the markets, or if there are defective or even counterfeit batches, which is the biggest problem that we're facing at the minute. And the OMCLs also provide analytical backup for the regulatory authorities. So if there is a problem which is identified with a vaccine, more often than not they will be sent back to the OMCLs, and they will perform tests or additional tests on products. Examples of this can be things, I don't know if you remember the Northwick Park incident, where there was a clinical trial where there was a number of patients. Well, that product came back to NIBS to check that everything was okay with it, and we actually did quite a lot of research on that product and identified the reasons why the results were as they were, and identified the flaws in what was done. So that's the other roles that we use. I haven't provided any handouts, but if you do want more information, then have a look at the NIBS website, or there's also more information on the EDQM, the EMEA, or even the WHO website, which talks about the regulation of vaccines. That's all. Thank you.